Pleased to introduce Dr. Aijung Wang. I think most of you know Dr. Wang. He co-directs our bioengineering program here, but in addition, he recently was awarded a Dean's Scholarship for Entrepreneurship and is helping to sort of educate all of us, learn himself, and then educate all of us on, sort of how to move new developments, new ideas, whether they be mobile device things or discoveries in the laboratory into an actual product that can help patients. So we look forward to your presentation.
Thank you, Dr. Farmer. All right, thank you. Good morning. So is this good? Can you hear me okay from the back? All right. So every time I present here, I feel, well, it depends on the day. Sometimes I feel very different because what I do is not directly associated with the patient care, like what we just talked about in M&M. Sometimes I feel better because what I do can potentially help. So today I'm going to, as Dr. Farmer mentioned, I'm going to give an overview of the research in the lab and also how we potentially can translate what we develop in the lab into commercialization product that we can use in clinic. So it's not going to be happening tomorrow, but will be near in the future. So this is the outline of my presentation today. So first of all, I will overview what we're doing in the lab, just to give a introduction to anyone who is not very familiar with the lab research too, and or anyone who is interested in knowing more about the lab, what we can offer. So the second part is I will give some introduction to a few examples of innovation and invention that what we're doing in the lab, a case study. So the last section, I'm going to try my best to talk to you about what I've learned so far, and also what's going on, not just in UC Davis, but also other universities and other places in the world. What surgical bioengineering can actually help in terms of innovation and medical device and stem cell products commercialization potential. All right, so surgical bioengineering lab. So as you may know, I'm a PhD in biology. I was trained, so right before I came here, so I was trained as a postdoc at Berkeley Bioengineering Lab, Bioengineering Department. So from my training and from my graduate school study until postdoc training until now, so what I'm really interested in is tissue regeneration. So we're interested in translational regenerative medicine. So we want to try to do translational research, meaning that from the bench to potential to bedside. So translation from the lab research into the product that we can use for patient care. So we're taking this from two main angles. Number one is stem cell engineering. The second one is biomaterial engineering. The reason we're doing this is that if you really think about the whole body and especially regeneration process, actually it's about cell and stem cell especially function. And on the other side is how the cells is functioning especially through the environment. So medical device and other supporting device system can actually help the stem cell or cell function behavior to get a better healing and better regeneration. So that's why we're interested in using materials, biomaterials, to generate and to create structures and devices that can help the body to heal or the transplanted cell behavior. So overall, the research is driven or is led by surgical questions or surgical need. So I'm just trying to cover a few just from our research, ongoing research, and how the question actually is from. So apparently we have lots of unmet need from pediatric and fetal patients because the big farmers, they're not really developing lots of devices or treatments for these babies just because of the market. So we at the academic institution and in the lab, we're very interested in developing devices and treatments to help these patients who are younger and very small in size and at age. So we, as you all know, we do a lot of research about pediatric and fetal patients. I'm going to talk to you a little bit more later. So another example is plastic surgery, plastic reconstructive surgery. So apparently lots of the treatments and medical devices can be used in this field to help healing and regeneration. So burn, of course, so skin graft and lots of other materials, and also healing, wound healing, scarring, how stem cells and medical devices can actually help, and how we can improve the currently available devices is actually so we're very interested in this as well. So trauma, for sure, and this is what we're really good at. Just one example, like if spinal cord injuries spinal cord get trauma injury, or brain tissue got injury, so a lot of tissue will be damaged and lots of neuronal loss will be associated. So can we do anything to help these neurons or many other cell types to regenerate or being protected from injury? So that's something potentially engineering approaches can help as well. So vascular stuff is something that I personally have been interested in for years. So apparently there are lots of need. So because the increasing prevalence of cardiac diseases and coronary artery disease, for example, and many other like diabetes and a lot of other issues causing vascular problems. So hemodialysis is another example, being a very important part of transplant division. So all of these are examples. I'm hoping, that's my vision as well. So down the road, when the lab is more mature and developed, we're hoping to help every one of you in any way that you want to improve your practice, or if you're interested in innovation or invention. So the lab is there. But I want to give you a little bit educational information that I learned over the years, actually, how innovation and invention can happen, actually. So if you think about innovation, the process of innovation. So we always, especially from our department over here, so there are different approaches. I'm talking about what we're doing now here. But there are other approaches other places are using, for example, from an engineering environment where there's no medical expertise. So the starting point could be quite different. So what we start is usually from here. So we start from identifying a clinical problem, because that's what we do on a daily basis. And this is what we are good at. This is very important. Not everybody has this kind of resource. So as I mentioned, like from an engineering environment, so what they really need actually is somebody who can give some advice, some suggestion how their technologies or approaches can be applied in clinical environment. So we are lucky that we start from the most important point, the clinical need, clinical problem. So once you identify a problem, the next step you should be thinking about, so what's the potential solution for this problem? Can we do anything to solve this problem? It can be a new problem, it can be an old problem. It can be something that a brand new idea you want to try, or it's something that you improve from what you already have. So then the next step should be de-risk of the potential solution. De-risk, by saying this, I mean you can have lots of ideas of your solution, but not necessarily every single one of them would work. So you have to justify, at least from your own perspective, how actually this solution could potentially work in the real case. So you need to collect a lot of preliminary data to support your own hypothesis or your idea. And then you need to do some bench testing and prototyping. And eventually, actually, you probably want to test that idea solution in a model system before you really move into a human patient. So animal testing in some animal models will probably be very helpful. And then if you have all these done, And if it works all the way through, you could potentially think about how to develop this into a product. So this is actually the most tricky part. So, and lots of ideas are actually very great ideas, and you have data, but how this technology or idea can be really further developed is a tricky portion. And also after that, if you have a product or prototype and then a prototyping product developed, how you can really move this forward into a commercialization pathway. That's actually the most difficult part, which we're not very good at, but we're learning. So if you really think about just from a patent application or patent protection point of view, all these activities can be divided into three parts. Number one, so when you have idea, you have a potential solution, so to solve a problem. So you should file a record for innovation. So what is that? So I'm going to talk to you about that in a little bit. So overall, generally speaking, it's something to show, to prove if that's needed in the future, that you had this idea in the beginning, or when was that actually happening. And then after that, once you have the idea, you have the solution, you're very excited about this. After you file this record innovation, you should execute all these activities and generate data. So, and then that's the patent application process. So you have the idea, now you have the data that you generate yourself to support that idea and the solution. So that will give you the protection, actually. And then, at the very final step will be commercialization. That's actually, it's supposed to be the biggest part of the whole process because it will take a lot of time, resources, and expertise. So what I'm going to focus today is on this earlier part because, down the road, actually, you can participate, you can lead, you can work with a lot of other people to get this, the whole thing really happen to the very last stage. Okay, this is the record of innovation. So, every one of you, if you have some idea, you identify a problem, but you have idea how to solve this problem, you could do this, actually. You could really do this to prove, well, you don't have to go all the way through to the very end, but you can do this. So it's just to show that it's a record of invention that you can file through UC Davis Innovation Access. So what it looks like is like this, actually. So this is a form, we call it ROI form, that you need to have your name, your department, and then, well, if there's anybody who is a non-UC will be there. And then you need to provide some description of the invention and what it is, and some detailed description of the technology. And then you need to say why your technology is better than what's available right now, or why it is not available. You should describe that. And then who has been supporting you to do this. And then if this invention associated with anything outside of UC Davis, like material transfer agreement, based material resources that you use. So you just need to provide all these information. And then this is the most important thing, I think, for the whole form. When did this happen, actually? This is you're going to find out this date on the final patent being the priority. Well, not necessarily, but if you need this date, actually, for a future any patent-related legal discussion, this is very important to have. So that's why actually keep a very nice and clean record of your work is very important, because this is all the evidence. And then you need to say when actually this first test was done successfully. And then if you have any publication, okay, this is important too. If you want to file a patent eventually, you need to be very careful with the timing, the timeline for giving a talk and publication. If you give a talk, which is considered as a public knowledge after the talk, so then the novelty of the patent is gone. So that's already a public knowledge. You can't really file patent anymore. But if you file this first and then give a talk and publish, that's fine. So you have patent, both patent and your publication, if you will, like even the subject abstract. So, and then you need to provide some references just for the office to evaluate if this record of invention is worth of further investigation. So over the years, actually, we have the experience really. So this is on the time. So this is the earliest. This is the latest. So this is the earliest patent that we filed since 2013. Actually, we filed this 6. Actually, the first one is recently granted, being a patent. So how it works, the whole thing works is that once you have this filed, or you've submitted ROI, the Innovation Access Office colleagues will come to you, contact you, will talk to you about this potential invention, and you're gonna work with them and back and forth and describe the need and potential, lots of other things. And the first step is a provisional patent application. If this is really something they're really interested from this institutional perspective and also from your own passion, this is something that potentially will work. So university will help you file a provisional patent. And then you move actually to the PCT international filing, and there will be many cycles of review and your discussion with the attorney and the patent office. Usually it takes about 2 or 3 cycles. So eventually we'll come to a real patent. So that's a real deal with your name and date, what the claims are included, all these information. I'll show you very quick on that, what it looks like. But overall, I mean, it would take a long time actually. So what we have, Dr. Farmer and I were the author or inventor on the first one, actually took about 5 years. So it depends. Sometimes it takes longer, sometimes it takes shorter, but generally speaking, quite a few years. So I will give you 2 case studies, just very briefly, because I don't want to, so I can talk to you a lot about the details, but I want to give you an idea from a storytelling. How does this happen really? If you have an idea, how can you really do this until you get the patent? So I'm going to give you 2 case studies, just based on this one example from each direction that we will be focused on, stem cells and biomaterials. So the first one actually is the stem cell part. So as you may know that we actually have been working with stem cells for the whole time, and we're focused on 2 major things about stem cells. One is that so stem cell sources, which means where can you generate the best stem cell for certain application. So it can be any tissue, that donated tissue or available tissue, or any engineered cells from available samples of tissue, like induced pluripotent stem cell differentiation. So what we actually have been focused on over the years is the perinatal tissue, which usually is discarded. So there are lots of stem cells from the perinatal tissue: umbilical cord blood, umbilical cord, placenta, amniotic membrane, all these stuff. Usually, I mean, there are lots of good stuff there, but unfortunately, it's really underutilized. So we have stem cells, and we actually are developing stem cell products. What do I mean by that? So stem cell products, I want to emphasize the off-the-shelf capability. So it's a product. It's meaning that it can be used to different patients. It's off the shelf, and so which means allogeneic cells actually, which is very widely used nowadays. And also potential for a cell-free product. So stem cell or live cell derived product, but it's not live cell based activities. So it can be conditioned media or secreted stuff from the cell, like exosomes. We're interested in nanoparticles secreted by stem cells. All these things can be collected, isolated, and can be stored and can be used as to substitute stem cell functions. So that's the stem cell part. And the second part, I will talk to you about that in a little bit. Very brief, this is how the patent actually looks like. I don't know if Dr. Farmer has seen this. This actually has recently been granted. It's less than a month ago. So this actually takes a long time. I want to show you this is actually the priority date actually was in 2013. That's when we first filed this patent. Took quite a few cycles of discussion with the patent office and review. And well, first of all, you get in line actually to be reviewed. It takes a long time for that too. But anyhow, so eventually actually we have a patent that can protect us so we can be very protected to carry out this work moving forward. So the first patent actually is about the pediatric application, as I believe everybody in the room knows about spina bifida, because this is really something we've been working on extensively. So spina bifida is a birth defect. It happens very early in gestation, and it's from a lot of basic research, we know the reasoning of the defect, or what happened actually early in gestation. It's about cell migration actually, because it's opening usually in lumbar area. So the spinal cord is exposed versus, normally it should be covered and protected by a lot of connective tissue. So from a lot of basic research, and especially the clinical research, for example, the MOMS trial that Farmer was a PI on, supported by NIH. So all these studies actually indicate that spina bifida is not something that is untreatable. It is something that we can actually help. So that's why we're looking at the surgery plus something else. So we have done a lot of surgical treatment for spina bifida, but the question was, can we further help these patients by providing some biologically active components, for example, stem cells? So why stem cells? The main reason is that, for spina bifida and many other developmental problems, stem cells are a very key factor over the process, because stem cell activities are very active over the gestational period. So why placenta? Actually, before placenta, we've done a lot of research about other stem cells. So over the years, we came to this point, like placenta. So the main reason why we're using placenta, number one reason is the availability. So availability of the tissue, and also potential availability of the cell derived from the tissue for autologous treatment for the patient. But not necessarily we have to do that. Once you have the cells, you could potentially use those cells allogeneically, so from one donor for other patients. So placenta is available across different gestational ages, term, preterm, and even very early gestation by chorionic villus sampling, for example. And so why placenta again? Placenta as an organ is very important for development. So as an organ, actually, it functions to protect the fetus and also to provide a lot of reagent, a lot of very important factors for the fetal development. So we, in our lab, have developed a protocol to isolate a population of stem cells called mesenchymal stem cells from placenta tissue. So it's very unique, actually, this population of stem cells. So number one thing is that it's highly expandable. So from a very small tissue, for example, from a CVS, chorionic villus sampling, it's only about 50 milligram, 100 milligram tissue. So over 20 something days, 4 weeks-ish, we can get a billion cells, actually. It's a large number of cells. And you can actually get more than that by doing different culture protocols. We're optimizing this culture protocol. So the idea is to get enough number of cells for a autologous CVS patient, so biopsy placenta, over a couple of weeks, 2-3 weeks, you have enough number of cells for the same patient. So the same technology can be utilized to donor-derived tissue, which can be much bigger than this. And you can get a larger quantity of stem cells, which can be stored, can be banked, which can be used for a lot of other patients. So there are lots of other unique properties of these cells. They're very important. They have important development-related functions, which other adult cells or adult tissue-derived cells do not have. And they're fetal in origin. So if you really want to emphasize that, actually, that's something very unique. So I don't want to talk too much about basic science today, but I want to show you. So once we have the cells, actually, we have the identify a clinical problem, and we have a solution, stem cells. Now it's coming to test these solutions. We have tested these products, stem cell products, in different animal models. This is one of the most well-established model, the fetal lamb model of spina bifida. So what we found in the study, actually, by supplying stem cells with fetal surgery, actually, as a combination, so they can rescue motor function for the fetal lamb with spina bifida versus the lamb without stem cell treatment. Actually, they will be paralyzed. The lambs treated with stem cell, they have normal function at birth, motor function, walking and motor, or other aspects. From histology, we identify, now we understand more and more the mechanism of action. So the stem cell can rescue a lot of neurons, which actually can be applied to a lot of other neuronal diseases. If neuroprotection is a mechanism, our stem cells can potentially protect neurons in this particular environment, but potentially can be applied to other environments. So recently we are starting this vet school collaboration by using a naturally occurring disease model of spina bifida. So versus the surgical model, this is a naturally occurring disease model using the English bulldog who developed spina bifida naturally. And we actually found this is very similar from human point of view, and this is the dog version of spina bifida. It's similar, has a lot of similarities, and we're actually doing more and more dog treatment. I believe I presented this lots of videos last time, but we're getting more patients done. We're very excited about this. So all these, you need to have some support. Actually, it's lots of the animal studies are very expensive. So we're lucky that Dr. Farmer and I are co-PIs on a grant supported by CIRM, is called Preclinical Development Award. So Preclinical Development Award is actually in the later stage of the translational grant, actually. Now we're talking with our GMP facility. We're really talking about product development, actually, at the GMP, good manufacturing practice facility, which is required for a product that can be used in human, like a medical device, drugs, and stem cells. Stem cell products need to be produced eventually for human patient use. You need to produce these products in the GMP facility. So we recently submitted this grant to CIRM. This is the Clean One. So it's the next step after preclinical development. So it's the late stage of preclinical projects. So we're talking about GMP production and qualification of these cells for future potential human clinical trial. So hopefully we can get this, and we're really going to be moving this technology from the lab, from the animal model to patients, animal patients into human patients. That's our hope. Hopefully in a short period of time we can report to the group that we're getting ready. So the next case study is on the second direction, which is the biomaterial direction. I have to go through these slides a little bit faster because there are lots of technical issues or technical aspects of that. So this is the patent application that we filed a couple years ago on this research. I will talk to you about the details. So again, the problem comes from a clinical need. Cardiovascular diseases, as you know what I mean, right? The unhealthy lifestyle actually is changing a lot of things, especially in the cardiovascular disease field. So in a lot of cases, when the blood vessel, for example, is really bad, that you need to replace it, so a graft is needed. So either being a autologous graft from a vein that can be sacrificed or has to be sacrificed, or off-the-shelf artificial grafts that can be used to replace and to refunction the location. So however, I mean, all these off-the-shelf products actually, especially PTFE graft is widely used in clinic. As many of you know that, so these grafts have no regenerative potential. So they are Gore-Tex material that on your jacket, actually, they're put in the human body. It works very well, actually, in rescuing a lot of functions, but it has a lot of disadvantage of this material. So how can we improve this product? So lots of research is going on in this field, actually. So apparently there's a large market for this, large market for vascular graft. So there are about $2 billion. I couldn't find the most recent, but 2014 is about $1.95 billion market. And there are a lot of reasons that causing the increase because of rising prevalence of a lot of diseases. So for all these artificial grafts, actually, if you really think about it, it doesn't have any regenerative capacity. It doesn't have any biological function to start with because the PTFE material is synthesized and it doesn't have anything. So if you think about all the blood contact environment where the blood is going through, and if there's no endothelium there, actually, it will cause a lot of problems. So a lot of other cells other than endothelial cell will bind to the surface, cause a lot of infection, a lot of thrombosis, a lot of other problems. So endothelium also functions like a barrier, so to inhibit binding from other cells, and also a modulation layer of cells and crosstalk with smooth muscle cells underneath. It's very important. If you don't have it, you have a problem. You better have it. So that's the justification. And so we want to generate endothelialization on the intravascular or intravascular devices by modifying the surface. So how can we do that? So in circulation, actually, there are lots of cells that are called endothelial progenitor cells. Not very many, but they are there. So if you don't utilize them, they can't be functioning in the way that you want them to. So, but you can engineer the surface of the graft to provide a binding site for the cells. When they're going through, actually they can be captured and they can. So that's the initial idea. We focus on integrins because integrins are very important for cell binding, migration, as we all know, right? From basic biology, integrins are, and different cells have different integrins and different situations, different environments actually will cause cell to express different integrins. So you know all these, but what's the solution? So the idea, so you identify problems, and you have so what's the solution? So our solution was to utilize this endothelial progenitor cells that are rare in the circulation, but they are there. So if you can use these cells and screen a binding site, a binding ligand from a high throughput screening system, for example, one bead, one compound, I talked about this in my previous slide, just for people who do not know this. This actually was invented by Dr. Kit Lam on campus by chemistry. So we collaborated with Kit Lam's lab, and we used endothelial progenitor cells and endothelial cells as a targeting cell type. And we utilize the integrin expressed on endothelial cells. This is what we're looking for. So this is the solution. We're looking for, so there are other ligands on the market actually, or in investigation. So what's making your idea or your solution unique? So by using 1B1C, actually, you can find higher specificity. So binding ligand that can more specifically bind to a cell type that you're very interested in our case, EPCs. And higher affinity, so capturing capacity. So it binds very, very tightly, very sticky. When the cells go through the vessel, it won't go by, it will be captured. So that would be ideal. And then stability, because there are lots of native amino acid-based sequences, like peptides, native peptides and proteins, they're not that stable. So for future applications, it can be very problematic. So which means, like growth factors, you have to apply that like more frequently. So, but in our system, because it's so engineered, everything is artificial, actually, it's engineered. So we can use non-natural amino acids for the building blocks to make that structure very stable. Of course, you have to meet all these specificity and affinity requirements. So eventually, actually, we identify lots of ligands. So I don't want to go through all these. So we find some ligands, actually, they have different binding specificity. This is endothelial progenitor cells. We call them ECFC, endothelial colony-forming cells. Is there colony forming there in the stem cell progenitor cell stage? These are human coronary artery endothelial cells. So they're mature endothelial cells. You can see this ligand, for example. So EPCs, endothelial progenitor cells, can bind to this ligand very well, but mature endothelial cells don't bind to that. This is because the integrin expression on different stages for one cell, but they express different integrins. So that can be utilized. Actually, this is alpha-4 beta-1 integrin. So this is THP1 monocytes. We don't want to get too many monocytes to the regeneration site. Well, we don't know about that, but at least at this very much, like not very well known, but at least at this situation, we want to have more endothelial cells. So this is ligand actually we identified. It functions very well for endothelial progenitor cells and also for mature endothelial cells, but not for not for monocyte. This is just one example. You can think about the potentials. A lot of potentials. You can find a lot of things. So we also did so basically I want to just in a very short summary, this ligand actually binds to this EPC and ECs very quickly and very strongly and very specifically. But that's years of work, just summarizing in one sentence. So to see what's the function, so you can imagine you can capture something, somebody, some cell, but what's the next step? Is the cell will be killed by the capturing process? Is it too tight? Would cells be trapped and would not be able to do anything? But actually, no. This is the beauty of this ligand. Actually, we've done. That's why it's summarizing this slide. Actually, is years of work too. So, as you can see here, EC attachment with the ligand treatment is significantly higher than the no treatment group. And this is the important slide. Actually, hopefully you can appreciate that. So over the culture time, with the ligand, actually, the endothelial cell proliferation is increased. What does this mean? So endothelial cells are going through the vessel all the time. Some of them are captured. But once they're captured, actually, they are able to be proliferating faster. They can, so in an ideal world, if you really don't think about the bad side, but potentially they can, when you need a lot of endothelial cells, and then the endothelial cell can be captured and they can proliferate, they can cover the exposed area quickly. So that will actually diminish a lot of the potential risk for not being covered in the bed of endothelium, but being regenerated by endothelial cell will be changing a lot of things. And also, very importantly, the ligand can actually increase the biological activity of the cells, like VEGF receptor phosphorylation, which is very important for a lot of functions of endothelial cells. So all in all, this ligand is pretty good. So that's our solution. So I have an idea, we have the solution, we found this ligand. So and we generate some preliminary data in the lab, but can this really be used for anything? So that's why we actually carry out lots of studies, including these three. So I'm thinking about this from the clinical use point of view. Polyester, polymer-based devices and scaffold, for example, polylactic acid, like our suture, biodegradable suture, and many biodegradable medical devices can be made by polyester, and more actually, because it's biodegradable. Once it's implanted, the heating process and the degradation, in vivo degradation process, will work together. So eventually, you don't have to take it out. It will be replaced by bodies, so by body tissues. So this is actually more and more biomaterial. It will be developed in like polylactic acid or polyglycolic acid, all those kind of polyester-based material. So this is going to be, more and more devices will be, can we use our ligand to modify these, or can we use like our technology to improve these devices? Second example, like SIS, Dr. Boyd is here, and lots of other decellularization process-based material, like alloderm, for example, that we use all the time. But and they're collagen-based. We have a strategy to improve endothelialization or vascularization by using our peptide. I will show you in just a second. And also the medical devices like PTFE devices, stents, metal stent, valves, material, catheters, all those hard to modify. All those kind of things are very synthetic and very hard to modify. Can we use our technology to do that? And the answer is yes. So I'm going to show you very quick about what we did for this. So this is the artificial extracellular matrix made by electrospinning. So the nanofibers, because native ECM is made of nanofibers, so we can make all these by polymer, actually. It's a polylactic acid based. So we're making these, but these, there's no binding site on this polymer if you don't do anything with them. They are just polyester. Cells can sit on them, but they don't really react with them. They don't interact with them at all. So that's the idea. So we have some chemistry-based approach to put the ligand on the surface of this, if you really think about from molecule level. So on the surface of the nanofiber scaffold, we have the ligands actually modified on the surface by chemistry. So we showed that confirm the presence of the ligand on the surface, and this is the important slide, just for interest. With the ligand modification, compared with the untreated surface, actually, it's not very clear, but you can see a lot of endothelial cell. This is endothelial cell seeding. Endothelial cells can be captured and bind to the surface very efficiently. And so if you do a short-term binding and wash off the cells that cannot bind, you can see that X7, this ligand can increase the binding very efficiently. Well, it's cut off, but this is actually the covering area, so which is very important for if you think about endothelium on a blood vessel bed. So the coverage overall with the ligand, actually, after 2 days of culture, the coverage area percentage is much increased compared with the non-treated group. So this is an in vivo model. So we use a rat carotid artery model. And with the ligand modification, actually, you can see the patency rate for these very small diameter vascular graft is about 0.9 millimeter. It's about 1 millimeter, actually, 0.9 to 1 millimeter inner diameter. It's super tiny. And we use this purposely, because if you don't do anything for such a small diameter vascular graft, it can be easily clogged over time. And with the modification, as you can see, actually, the patency rate is very much higher, especially, I mean, we have about 6 weeks. And so this is what happened, actually, in vivo. You can't see all these details, but I want to tell you, with this ligand, this is the middle of the graft, actually. This is the proximal, closer side to the heart, and distal. So with the ligand, actually, within a week, at 1 week time point, you see a lot more cells that are captured in the middle of the graft versus in the untreated surface. You don't see much cell population there because there's no reason the cells can be captured. So with the ligand, actually, a lot of cells, they are positive for CD34, which is a marker for endothelial progenitor cells. Over time, if you skip this one to this one, so from 1 week to 2 weeks, a lot of these red cells, which is CD34 positive, they become into green cells, or more green cells arriving. So they are mature endothelial cells, more than the untreated surface. 6 weeks, actually, you have a relatively mature endothelium. So that's pretty striking. Within a very short period of time, you can see a large coverage and lots of endothelial cells in the middle of the graft. It's about 1 centimeter in length, so it's in the middle of the graft. Usually, it's very difficult for endothelialization to happen. So with this ligand, actually, you can see a lot of endothelialization. So the second example, I will be very quick on this. So SIS and this one, PTFE. SIS is a collagen-based material that we use a lot for a lot of clinical applications and lab applications. And alloderm SIS, for anyone who uses this, so I think it's actually already a very good product. So, but one thing that we can potentially improve on this product is the potential survival potential for alloderm, for example, and also the vascularization. If you can supply more blood vessel into the structure, that can help the integration of the transplanted device with the body. So by supplying a lot of blood supply and also nutrients for the regenerated tissue. So by using this ligand, actually, this is SIS. So this is SIS plus ligand. So by using this modification technology, so this is endothelial cell seeding, very straightforward way to see this. So without coating, actually, it's not bad. So endothelial cells can adhere to the surface, but with the modification, you see a lot more cells. And 30 minutes, actually, you see a very much increase in cell binding on the surface. Over days, actually, 1 week and 3 weeks, actually, you can see some mature vasculature in the SIS scaffold by this ligand. It's better than the scaffold without modification. But as I said, SIS is a very successful product. It's widely used in clinic. There are lots of other products, especially for thicker material, that vascularization is more important, actually. We're doing a lot of other things. For example, bladder matrix, so decellularized bladder matrix for bladder tissue engineering purpose, for example. So if you don't have any binding site for endothelial cells don't like to bind to them. Because there's no need, there's no incentive for them to bind. But once you have the modification, the endothelial cells and vascularization can be actually improved quite significantly. So this is the last example for any of those hard to modify material, PTFE graft, metal materials, valves, catheters, all these materials, actually, we can modify these surfaces by using our technology. This is a PTFE graft. This is a PTFE graft. This is a culture dish. So this is regular coating with our control. This is a PTFE graft with our ligand coating. You can see within 10 minutes, actually, this is a very initial study, but it showed the promise of this coating technology of promoting endothelial cells of attachment to the surface. So as you can imagine, so if we can do this technology can be applied into a lot of medical devices if you want them to be covered by endothelial cells, populated by blood vessels. And so you can think of a lot of applications. So that's the patent application. And so, okay, this is my last section of slides. So there are lots of research activities in the lab, and we can do a lot of things. So, but currently, actually, there are a lot of challenges and opportunities. Very quick, as Dr. mentioned, I was honored to be the Dean's Fellow in Entrepreneurship. The Dean's Office actually is very interested in these three areas this year: entrepreneurship, informatics, and precision medicine. So entrepreneurship actually is different from conventional academic activities, as you can imagine, right? But apparently it's very important. Innovation is required and more required in many areas. And this actually is the big idea is ongoing right now. So the second round. So our Chancellor, Dr. May, actually, this is the areas where it highlighted. So innovation and entrepreneurship being one of the 5 topics actually we're promoting this year. So as you can see from school level, from our UC Davis institution level, entrepreneurship, innovation is actually being recognized as a very important part. This is something I want to talk, just one very quick. So this is the federal funded short course. It's called I-Corps. So NSF and NIH actually, they're supporting this. So the idea is to train physicians and anyone who is interested in doing innovative research and commercialization of an idea or product. So it's a short course in a couple of weeks. And Dr. Jim Kovac from our side, actually, this is the first year UC Davis actually is participating in I-Corps. There are many other sites, but on the West Coast, actually, UC Davis is #1. It's ongoing, actually. I think if you're interested, you should reach out to Jim Kovac and let him know. So I'm going to skip this. Okay, Eggy Square. So this is actually, I saved this to the last. So Eggy Square actually is a big idea. I mean, I don't know if you know about this. It's really happening now. So it's UC Davis Sacramento Invention Center. So the Eggy Square is right here. So it's talking about us, actually. So I was honored to be on a team with Associate Vice Chancellor and many other, like from Office of the President of UCOP and other UC leaders to Georgia Tech. So Georgia Tech, the reason we're going there for Eggy Square is because Georgia Tech is very successful in making the incubator and the Tech Square, named by Tech Square there, and our name is Eggy Square, actually. Our Chancellor, Dr. May, actually was the Dean of School of Engineering, as you may know, at Georgia Tech. And over the years, actually, he was part of the leading team for Tech Square. It was very successful so far, has been a great story. So it was famous because it's an academic center-based incubation site for a lot of engineering technology development center. So it has been a big deal. And this is the 2001, I took this from the internet, actually. I have some slides from the trip, but I couldn't really, but this is the similar thing. Idea is that, so about 10, actually it's about 15, 17 years history. So Eggy Square, this is 2016, actually. But now more and more activities are ongoing. It's a huge change to the community and a big academic and economical advantage to the whole society. And there are lots of incubator properties there. And this is, well, to the campus and to the community and also beyond and to the world, actually. A lot of these innovations and big companies actually go into that area for further development and technology commercialization. Okay, so just to conclude, I hope I didn't go too fast, but I want to emphasize that surgical bioengineering innovation and invention, they can really mature into applications, future treatments. And what we are hoping to do now actually is to establish the surgical bioengineering lab as a hub for innovation and invention and education to anyone in the room, anyone on campus who is interested in surgical bioengineering innovation. So we want to integrate all these robust collaborations and all the resources on campus at UC Davis, the vet school. We have the best vet school. We have a very nice engineering school, and we have us. So all the collaborations can potentially help the generation of new ideas and new technologies. And we would also like to offer the mentoring, experience-based mentoring, for example, to any physicians, residents, scholars, graduate students, medical school students who are interested in inventions and innovations. And there is definitely a big opportunity right in front of us. And thank you very much for all these funding support for our research. And this is the lab who did a lot of the work that we have presented. And thank you very much for your attention.
Well, thank you, Aijung. We're a little bit over time, but I may just take a minute for one or two questions. Ian.
First of all, thanks. I really appreciate your talk. You made me very optimistic about the future. I was just thinking you very well went over the evolution from scaffolds that just are inert to scaffolds that bind to scaffolds that differentiate. And I was wondering if you were thinking about bioactive scaffold ligand combinations that actually promote directed differentiation and marrying that with your stem cells.
Right.
That's to make it like exactly what you want it to be. So it's not only binding, but it's actually causing.
Absolutely. So definitely a very important point. And to guide the fate differentiation of stem cells, instead of making them, to do anything they want, or, that's the hope. So we're interested in that very much, actually. One of the examples, we're doing a lot of things around that, just to precisely, or as precisely as possible, that we can do in the lab, to control the cell or to help the cell go to the direction that we like. So integrin actually is one of the molecules we're interested in, actually. For example, alpha-4 beta-1 integrin is known, very important for homing of stem cells from circulation to an injury site. And after that, actually, the downstream, after they recruited, they're going to be guided to differentiate or to do something. So we're trying to replicate and to use that knowledge to engineer the scaffold for culture and for transplantation. That's a good point.
Next level,
yeah.
I was just thinking of the stents that we're talking about in the GI tract or other places. You could think about lining those as well. We're not there yet.
Dr. Ali?
I thank you very much.
Thank you.
So I'm sort of thinking about how surgery has come along. So when we develop the therapies and then we figure out how to make the therapies less invasive, right? It's sort of the way that surgery has gone. And I'm just curious, this is just your perspective. Where are we, or maybe is it too early to start thinking non-traditional, less invasive delivery option? Because there's a lot of very nice technology, but the delivery, and in my mind, I remember, as a kid watching and the nanites carry all of the things down to the tissue level and fix it all. Where's that technology? I'm just curious.
Sure. I'm not an expert to answer that question, but just will try my best. So, it's definitely very important and it's underdeveloped, and it's being very important for the field. For example, stent. It's more and more versus intravascular stenting versus open operation to replace the material. That's actually is one good example of how important this field actually is feeling about that. But it's tricky, right? It's not that easy. So we are hoping to offer, lots of these devices to be hopefully in the future, not now yet. So if they have the memory, so you can make them small. Especially for those all the very porous structure, the mass value actually is very small. You can make it small, and once you implant in place, it can enlarge. If, for example, some of them become into porous structure to allow cell migration and repopulation, those kind of things, that would be ideal. It doesn't really. So we're hoping we can make something like that in the future.
So I think, the next step for us is to figure out how to get, we've developed sort of our outcomes research group, how to get sort of our entrepreneur innovation group together to think about these things and just brainstorm what ideas all the time. And then we can, we can recruit our engineering colleagues because they don't know the clinical problems.
Right.
Yeah.
Set
that up well. And they're looking for problems to solve with all of their 3D printers and all of their other things. So we can help foster those relationships.
Absolutely.
So thank you again, Aijung.
Thank you very much.
We look forward to
All right. Thank you, everyone.